Hi, in this lecture, we are going to start our study of the respiratory system. So why do we have a respiratory system? Well, number one, cells have to make energy. They have to make ATP. We need ATP for our cell to do everything a cell needs to do. Normal maintenance, growth, defense, cell division, and we get ATP primarily through aerobic respiration. Now, you probably remember that the mitochondria was the organelle for this process, and that the mitochondria is going to constantly take in oxygen. And as it takes in oxygen, it uses that oxygen to produce ATP, which is necessary for life, but then it also produces carbon dioxide, which is a waste product that you need to eliminate. Now, cells can go without oxygen for various minutes. The cells that are most dependent on oxygen are the neurons. And a neuron can only go without oxygen for about 4 to 6 minutes before it's going to die. So in this lecture, we're going to do a real brief overview of functions of the respiratory system, but then we are going to delve into the anatomy of it. So functions of it, we have number one, pulmonary ventilation. This is a fancy word for breathing. Breathing is inhalation and exhalation, moving air in and moving air out. We have external respiration. External respiration is gas exchange that happens in the lungs. So we're looking at the gas exchange, and again, the main gases that we will be studying will be oxygen and carbon dioxide. And so these gas exchange processes are going to happen between those little air sacs in the lungs, called alveoli, and then it'll happen between the lungs and the blood. Now the blood will be, remember, in a pulmonary capillary, because that's going to be the blood vessels in the lungs. So we call this external respiration. And as you can probably imagine, the oxygen will go from the lungs into the blood, and the CO2 is the waste product. It will go from the blood back into the lungs. Internal respiration, on the other hand, is gas exchange that happens between our tissue cells, so all cells of the body. And this time when we're looking at blood, we're looking at our systemic capillaries. And in this case, we still have the exchange of oxygen and CO2, but now the oxygen is going to the cell, and the CO2, remember, is produced by the cell, by those mitochondria, and it's a waste product, so it's sent into the blood. We also know that the respiratory system has protective mechanisms within it. We know it produces sound, and our nose also functions for smell, olfaction. When we look at the organization, it's divided into an upper and a lower. And in this picture, you can clearly see the upper respiratory tract, and this is basically from your larynx up. And the larynx is a lot of cartilage. You've seen it on models. We'll be studying it soon. But we're looking at nose, nasal cavity, most of the pharynx. The lower is the area below the larynx. So you can see we're going to have our trachea, our bronchi, bronchioles, and also the alveoli. So we're going to have an upper and a lower. So if you have an upper respiratory system, infection, then we're looking up here, maybe a sore throat. If we're looking at lower, maybe a bronchitis. When we look at the tract, there's something else, another way it's divided. It's divided functionally. So there's something called a conducting zone and a respiratory zone. Now, these are not the same as the tracts, right, about the upper or lower respiratory system. The conducting zone is the area that only has air that does not participate in gas exchange. So it's coming in your nose, your pharynx, your trachea, your bronchi. It goes all the way down to structures called bronchioles, and they're actually called terminal bronchioles. So the terminal bronchioles are the end of the conducting zone. So the conducting zone is important. It helps to moisten, humidify the air, filter the air, warm it up. So it has important functions, but it never actually gets down in the alveoli, so it doesn't participate in gas exchange. So any of the oxygen that's in this area does not get into your blood. And so we call this area the anatomical dead space. So it's the air that's in the conducting zone. It's about 150 mLs, and it's just the air that you inhale and exhale. So you inhale and you exhale, and it just never makes it all the way down deep into the alveoli. Now the respiratory zone is where the gas exchange occurs. So this is going to exchange gases with your pulmonary capillaries. And this consists of structures called respiratory bronchioles and alveoli. And we'll look more at this anatomy shortly. We also need to review the mucosa. You probably remember pseudostratified ciliated columnar epithelial tissue. The cilia are going to move, right? Cilia move, and they're going to help move organisms up and out and clean your respiratory tract. Lots of goblet cells. These are the cells that make mucus. Don't worry too much about the lamina. That's just the connective tissue below. But the entire conducting zone is lined with this tissue. And in this image, you can see all the cilia. And so what happens is this forms part of our defense system. Those goblet cells make mucus. Well, mucus is sticky. So when you inhale, any debris, pathogens can get stuck in the mucus. And then the cilia only move one way. They always move things up. So they're moving everything up out of your lungs all the way up to your throat, your oral pharynx, and then you swallow it. And you swallow all that debris and it goes in your stomach. And a lot of it's killed by your stomach acid. So this is sometimes called the mucus escalator. And it's the cilia that are constantly moving and cleaning out your lungs. And then we swallow this. Every time we swallow, we're actually swallowing some of the debris that got moved up out of our lungs. We also know we have hairs in our nasal cavity that help to filter and remove larger particles. And then we even have macrophages deep in the lungs. These macrophages are called dust cells, and they patrol down in the alveoli to help gobble up any foreign pathogens. So if we look at the anatomy, it's pretty straightforward. I like this table. It's not from your book, but it's a really good overview of basically what you have to know for anatomy of the respiratory system. We'll be looking at these structures in lab, but I just want to go over some of this quickly. So obviously you have a nose and you have nasal cavities, right? We have a right and left nasal cavity. The nares are your nostrils, and you probably remember that the bulk of your nose that you think of as your nose is actually hyaline cartilage. Remember, the nasal bones are up high. So inside our nose, we have hair and mucus. Again, really important to filter. It moistens the air. It basically makes it very warm and moist and humid. And then obviously our nose is what helped us with smell. Now, you probably remember the sinuses. You learned about these back in A&P 1. So we have what are called paranasal sinuses. They're spaces inside the skull. They help to lighten the skull, make it a little lighter. They are lined with mucous membranes. And you probably remember there was a frontal sinus in the frontal bone, maxillary sinus in your maxilla, and then deeper behind your eyes, you had an ethmoid and a sphenoid sinus. Now, because these are lined with mucus, sometimes if you have excess mucus or it's blocked and it doesn't drain, you can get bacteria. It can be viral, but you also get bacteria in there. And the maxillary and frontal often get affected with what's called a sinus infection or sinusitis. Now, the nasal septum, on the other hand, is just the piece of bone that actually divides your nose into your right and left nasal cavities. When we look inside, so this is a mid-sagittal cut right through the head, so you can look inside. Again, in this view, we're actually looking at the left nasal cavity, and you can see the whole side here. But you have a hard palate, which is formed from your maxilla, and then you have a soft palate back here, which is formed from something called your uvula. Let me write that there. And the uvula is kind of like the thingy that hangs down the back of your throat, and it's part of your soft palate. Now, the other thing you'll notice here is there's something called nasal conchae. So they're concave when they're plural. You saw these a little bit in A&P 1. I'll go back to this picture. There's actually 3 here. They're the pieces of bone that stuck out into your nasal cavity, and they're called superior, middle, and inferior. In A&P 1, we only really saw the middle and the inferior on the skulls, but they're just bone-like shelves that stick out. And then between them are openings called meatuses. So a meatus is an air passageway. So what happens is when we breathe, you can see the conchae sticking out here, the air has to spiral through these. And so it's really important because it really helps warm and filter the air even better as it spirals through these meatuses. And you'll see shortly too that, we're supposed to breathe through our nose, not our mouth. Breathing through your nose is much better for you. But because there's so many blood vessels here, that if you were to have any kind of trauma or got punched in the face or hit yourself in the face with something, you can get what's called epistaxis. And this is a nosebleed. And if you've ever seen one, the nose can actually bleed quite a bit. It's very vascular because that blood is bringing heat to help warm up the air. Now, once the air comes in, so the flow of air, we go in our nose, we go through our nasal cavity, and we start making our way back to our pharynx. Now, the pharynx has 3 zones, and you can see it better in this picture. The blue is the nasal pharynx. The oropharynx you probably think of as your throat, and then the laryngopharynx is a little deeper. I like to use the uvula as my boundary. Remember the uvula, the thing that hangs down the back of your throat? And so I usually go anywhere above the uvula, superior to it, I would consider the nasopharynx. Right around it would be the oro. And then once you come down further, this little piece of cartilage you might know is called the epiglottis. It's a little flap. And then right below the epiglottis is where I would label the laryngopharynx. Now, if we're looking at the model to try to label some of this, this is one of our models we'll have in lab. So, and you might remember it from A&P1, but remember we have our frontal sinus and we can see the sphenoid sinus. You might remember the pituitary gland right there, and there's your sphenoid sinus. We have our nasal concha. So technically you say concave when it's more than one. We say concha if it's one. So you have a superior, a middle, and an inferior. I'll put a little U here. This is my uvula. This is the tongue, in case you were wondering. And so if I'm going to try to identify the areas of the pharynx, then I would find the uvula right here. And so this area above it would be my nasopharynx. This area here would be my oro, and then I'll draw a diamond. This would be my laryngo. So you can see the different areas there for your pharynx. In fact, this little blue thing right there, that's the epiglottis. So the air makes its way down. We go through our nasal cavity, through the 3 parts of the pharynx, and now we're down into the larynx. So the larynx is a lot of cartilage, and there's an opening here. So this is basically a hole or an opening for the air to come in. Now notice that the air comes in the back, right? Remember this was the tongue and your mouth? So when the air comes back, it has to crisscross and it has to go toward the front. Whereas any food that you eat has to go to the back because that's where your esophagus is. It's right behind the trachea. So because these kind of crisscross, it's really important that we have this little flap thing called the epiglottis, which basically will help close off the opening, which is the glottis, because we don't want food to go down into our trachea. So the glottis is the hole or the opening for the air. Now, if we were to look at the larynx, we know there's lots of cartilage and it looks kind of light blue on a lot of models and pictures. And so the cartilage is called the thyroid cartilage, which you can see is the bulk of it. You can see that on the front really well. If you look at the thyroid cartilage, there's this little thing called the laryngeal prominence. You'll see it more on men where it kind of sticks out in their throat. You might call it your Adam's apple, but you have to call it the laryngeal prominence. We have the cricoid, arytenoid cartilage, and epiglottis. So the epiglottis is the flap-like thing. Remember that closes off the glottis to prevent food from entering it. And then we have the arytenoid and the cricoid. So the cricoid you can see on the front and the back. And then the arytenoid is just a little piece of cartilage, and it kind of sits, let me get a different color here, if this will work. You'll see it right in here. And it kind of sticks up. I always think it looks like a talon, or it looks kind of like it's a little piece of cartilage that we can see. And so those are the main cartilages that we'll be looking at when we look at the larynx model. So let's go to that model next. I'm having technical difficulties for a moment. And what we're looking at is our lab model. And so when you look at this, you can clearly see that thyroid cartilage, really big. This is the epiglottis. It's kind of like this big flappy thing that you won't miss. This is your cricoid cartilage. You can see a little piece of it there, and you see it more on the back. And then these little things that kind of stick up are the arytenoid cartilage. And so if we were to open up, these little white things here are your vocal cords. And when we open them, you would see a space, and that's where your glottis is. So we know we got to be careful. We don't want food or drink or anything to get into our larynx. And as I mentioned, the food has to go to the back to go down the esophagus, and the air has to come to the front. And so aspiration is basically when food or water or even vomit accidentally come into the trachea, and they can get all the way down into the lungs, which could be pretty bad. I mean, you could technically, if you aspirated enough water, drown in your lungs, but it can cause infections and trouble. Where the Heimlich is when you have a piece of food lodged. So usually it's in the trachea, and then you would do the Heimlich maneuver if someone was choking, right? So you apply pressure, and it hopefully will help force that food out so you can restore your airway and restore the movement of air. Now, if we look at the larynx, there's two types of ligaments here. There's something called the vestibular folds. Now, the vestibular folds or vestibular ligaments are called the false vocal cords. They sit up higher, they're superior. The vocal ligaments are what we see on this model as little white flaps. And the vocal ligaments are your true vocal cords. So you can call them vocal ligaments or vocal cords. Either is fine with me. But this is what produces sound. So when we speak and we force air out that opening, that glottis, it hits these folds and it makes them vibrate, and that's how we get sound. So you can see here the glottis is open and here it's closed. And you can see the false cords are out here. Here's the true cords, and here's another picture. So if you were intubating someone, you have to go right in the middle there. You have to get inside the glottis. And so I'm sure you've heard of laryngitis before, and that's when we have an inflammation of our vocal cords, so they don't vibrate the way they should, and so it's very difficult to produce sound. So this can happen from viral infections, bacterial infections that can get in there, and it's just inflammation. Now, the air goes into the trachea. Oh, I did want to mention this bone right here. This is the hyoid bone. You can see it on the model, and you can see it here as well. So once we get from the larynx, so here's the thyroid cartilage, cricoid cartilage, we go all the way down the trach. Now the trachea is pretty big. It has about a 1-inch diameter, and it has C-shaped hyaline cartilage. So we see the cartilage on the front of it. The cartilage does not go around the posterior side. Can you think about why that might be? Well, if we come back to this image, here's the trachea, here's the esophagus. So remember, food is coming down the esophagus. And so we don't want that cartilage on the posterior side because it could interfere with the esophagus ability to open up if you're taking a larger bite of food. So as we follow the trachea, it comes all the way down to there's a piece of cartilage at the very bottom called the carina. It kind of has a V-shaped structure. It's just the last cartilage of the trachea. And right after that, it splits, and it splits into what's called the bronchi. So it's going to go into the bronchial tree. Now, sometimes if there's a problem with air movement, you might have to have a tracheotomy. And I just want you to know the difference. Technically, the tracheotomy is when there's a hole put into the trachea to form an airway. So if there's some sort of choking or blockage and you can't get air down to your lungs, you need to put a hole in there. And the tracheostomy is when you actually put a tube in there. So if we follow the trach down, it splits into my right and left bronchi. So bronchi is plural, bronchus is singular. So I have a right primary bronchus and I have a left primary bronchus. They enter at an area in the lung called the hilum. And you can see that a little better. It's just a little medial depression where all the vessels and bronchi enter and exit. And it's called the respiratory tree because it comes in and then it splits, and it's highly branched as it branches into smaller and smaller tubes. And so as it branches into things called bronchioles. And here's another view. We can see the trachea, the right primary bronchus, and we have the left primary bronchus. So if we're looking at these, there's actually a slight difference between the right and the left bronchus. Let me go back up here for 1 second. So when we're looking at these tubes here, the right primary bronchus, I don't know if you can tell, it's a little wider than the left. And it's actually at a slightly steeper angle. So this is important because if something gets inhaled, like maybe your child picks up a small pebble and they shove it in their nose, you're like, what are you doing? And they go like, nothing, and they sniff, right? It goes all the way down through the trach. It's probably going to end up in the right side, in the right primary bronchus. So typically that's the side that's going to be scoped first if they're looking for something. But then as we get smaller and smaller, we go into things called bronchioles. So a bronchiole just means that the diameter is less than 1 millimeter, and it branches and branches and branches all the way down, and you might see here terminal bronchioles. Remember, that's the end of the conducting zone, and then the next bronchiole after the terminal bronchiole will be the respiratory bronchiole. So in this image, notice you can see the bronchi. They're going to split. And what's interesting, again, why it's called a tree, is they split many, many, many times. And so what happens is the primary bronchus will come in, and then it will split. You know, I'm not the best at drawing. And so I'll just do this. This is primary. It'll split to form secondary, and then the secondary will split, and they're going to be tertiary. But then these will keep splitting smaller and smaller and smaller. And they actually say it splits up to 23 times, where the alveoli is the 23rd split. So that's pretty highly branched in your lungs. I wanted you to also see this picture. So notice this is your terminal bronchiole. Remember, this is the end of the conducting zone. The respiratory zone starts with the little tube called the respiratory bronchiole. And then there's usually, they kind of look like a cluster of grapes if we were to look at the alveoli coming off. So we'll have the alveoli, and usually there's a little duct there. Now, one thing we notice, look at the cartilage. As we go from big to small, the amount of cartilage actually decreases while the amount of smooth muscle increases. And the respiratory zone, if you look, there's no cartilage here. So the cartilage's purpose was to prevent the airway from collapsing. Well, now these smaller airways have less cartilage, so we worry a lot more about any kind of airway collapse, any kind of bronchoconstriction, because that's going to increase resistance quite a bit. And so we know in the lungs that we have bronchodilation and bronchoconstriction. We know that bronchodilation is caused by your sympathetic nervous system, and bronchoconstriction, when it narrows, is going to be by your parasympathetic. So we're coming back to sympathetic, remember, releases norepinephrine. And then you might remember it's a beta 2 receptor when we're looking at the smooth muscle in the bronchioles. So different receptors. And when it binds here, it's going to cause bronchodilation. Whereas the parasympathetic will use acetylcholine. It'll use that muscarinic receptor, and that's going to cause bronchoconstriction. So bronchoconstriction, as you remember from the cardiovascular system, that's going to really increase resistance. So that's going to decrease airflow. That's not really desirable. Bronchodilation, on the other hand, is going to open up, right? And it's going to increase airflow. So here's a picture of the respiratory tree, and I think you can see all the branches, right, that happen here. And so bronchoscopy is just a tool where they can go in and they can look inside. They usually use a camera, so maybe they're looking for tumors or an inhaled stone or pebble or something like that. So I found this clip of a virtual bronchoscopy. So again, you have a camera, you're going down, you can see the split into your right and left primary bronchus. And it's just a tool done to visualize the inside of the bronchi. So if we come back to the respiratory zone, we're looking at the alveoli, which kind of look like a cluster of grapes. We have the respiratory bronchioles that are bringing air to the alveoli. And then usually there's this little opening here called a duct. And these are the alveoli or the alveoli sacs. Now notice that they're very densely surrounded by pulmonary capillaries. Lots and lots of blood vessels here for the exchange of gases. You might remember that the exchange of gases here is called external respiration. We're looking at gas exchange between air in the alveoli and blood in your pulmonary capillaries. Well, we know that oxygen and carbon dioxide use the process of diffusion to move back and forth. And so it's very, very important that we have a very thin membrane. And so each alveolus, you can see it here. They're actually, the next picture is a little better, but they have simple squamous epithelial cells. And these are sometimes called type 1 cells. So you can see them in here, the little flat, simple squamous cells. Very, very thin. Now, you might remember that the capillary, right, blood vessels, capillaries also only have one layer of simple squamous cells. We also have some cells in here, they're called type 2 cells. And these type 2 cells, or these kind of orange looking ones, they make something called surfactant. And surfactant is a phospholipid, and it puts a little coating inside to prevent the alveoli from collapsing. So it's very important to make sure they don't collapse. And then we even have some macrophages. We mentioned that they were called dust cells. So if we look at this, we have a very extremely thin membrane for these gases to move across. And so what this is called the respiratory membrane. And so it needs to be thin so we have the exchange of gases, the diffusion. And if we were to look at this membrane, remember it has 3 pieces to it. So number 1, it's going to have the simple squamous cells, the simple squamous alveolar cell, right, of the alveoli. And then we also are going to have the endothelium of the capillary. I'll just draw it up here, which is also simple squamous tissue. So 2 very, very thin cells. And then where they meet, you have the basement membrane of both. So this must be extremely thin for diffusion. In fact, I read once that if you took a single piece of tissue paper, 1 piece of tissue paper is 20 times thicker than this. So it's very, very thin. If we look at the lungs, we know that both lungs are in the thoracic cavity, but each lung is in its own pleural cavity. So we have a right and left pleural cavity. The base of the lung sits on the diaphragm and it's kind of flat, and the apex is the pointed part and it's superior. And in fact, if you take a deep breath, it comes right up by your clavicles. When we look at the lungs, they're actually kind of curved. This is called the costal surface because it curves around the rib cage. Now, the right lung is divided into 3 lobes, while the left lung is divided into 2 lobes. The right lung has 2 fissures. So fissure is a groove. There's a horizontal or transverse fissure, and then there's an oblique. So we see 3 lobes here. We have a superior, a middle, and an inferior. But if we look at the left lung, it only has 1 fissure. It only has the oblique fissure. So the horizontal fissure is the fissure that's in the right lung only. And so the left lung is divided just into a superior and an inferior. So if we look on some of our models, we can see, you can kind of see the grooves here, right? You see the transverse fissure and the oblique. And on this lung model, on the left lung, you see the oblique. So you see a superior and inferior lobe. Notice we see the thyroid cartilage pretty clearly. You see the hyoid bone. This is the thyroid gland. You do see your trachea. And then we can notice we have a left primary bronchus and a right. And then where it splits would be the secondary bronchus. And in lab, it'll look better when you can pull this off, and you'll see the bronchi even better when you can look up close. And so we know the lung has this very, very elastic tissue called the stroma. Remember, elasticity means that if you stretch it, it will recoil. So it's very important that it recoils. And then we have membranes. We have our serous membranes. Remember that visceral means that it always attaches directly to the lung. So each lung has its own visceral pleura, which actually attaches to the lung. And then around the outside of it, right, lining the cavity is the parietal pleura. So the parietal pleura lines the cavity. Between them, put little X's here, is your fluid, which is there to lubricate and reduce friction. And so you might have heard of pleurisy before. This can happen if there's not as much fluid and the membranes are very dry and they rub against each other when you breathe. So it can be very, very painful. And then finally, we'll wrap up with the blood vessels, which you've already learned. We know the pulmonary arteries are going to come in. There's the hilus or hilum again, that depression. And they're going to come in and branch with the bronchi. And so as they come in, remember, they're carrying blood away from the heart. They're trying to get to the alveoli. They're going to branch into pulmonary arterioles and then go into your pulmonary capillaries. The gas exchange will happen here, and then once that blood becomes oxygenated, O2 rich, it'll leave through pulmonary venules and be collected by the pulmonary vein. Now, just a shout out, remember that enzyme ACE is also made in your pulmonary capillaries, and that was an enzyme that was very important when we were looking at the formation of angiotensin II. Remember that ACE is the enzyme that converts angiotensin I into angiotensin II. And that'll come back a little bit this unit, but definitely in the next 2 units. So that wraps up our first lecture with the anatomy of the lung and the respiratory structures.